Radial unit hypothesis. The radial unit hypothesis of cortical development postulates that the embryonic cortical plate forms from vertically oriented cohorts of neurons generated at the same site in the proliferative ventricular zone, called proliferative units, which consist of radially deployed progenitors in different phases of the cell cycle. Thus, each radial unit and that was Rakesh, 1978. Thus, each radial unit consists of several clones, polyclones, that produce postmitotic neurons which migrate to the cortex following glial fascicles spanning the fetal cerebral wall and forming ontogenetic columns. After arriving at the cortical plate, the later generated cells bypass earlier generated ones and settle in an inside-out gradient of neurogenesis. Thus, the two-dimensional positional information of the proliferative units in the ventricular zone is transformed into a three-dimensional cortical architecture. The x and y-axis position of the cells is provided by their site of origin, whereas the z-axis position is provided by their time of origin, figure 1.3. The radial unit hypothesis relies on the proposal that neurons comprising a given radial column are clonally related, and this idea could be tested experimentally with the aid of the retroviral gene transfer method for in vivo analysis of cell lineages in the mammalian brain, Sains, 1989. Use of this approach suggested that most progenitors originated in the same site of the ventricular zone remain radially deployed in the cortex. Furthermore, a number of studies in chimeric and transgenic mice have provided evidence that a majority of postmitotic, clonally related neurons move and remain radially distributed in the cortex. The use of the retroviral gene transfer method in the embryonic primate brain showed that even in the large and highly convoluted cerebrum, radial development of many clones is remarkably well preserved. The radial unit hypothesis provides a conceptual frame to understand how changes in the dynamics of proliferation, migration, and survival of newly generated cells in the proliferative regions translates into changes in the thickness or surface area of the cortex during individual development and evolution. The study of this developmental mechanism has been done mainly using transgenic mice and retroviral approaches in human primates. Based on these data on the time of cell origin and cell proliferation kinetics, we proposed that the number of cortical cells and the size and morphology of the cortex depends on the mode of mitotic division, symmetric versus asymmetric, the duration of the cell cycle, and the degree of programmed cell death in the proliferative zones, as is described in the next section. Since most genes involved in cell production and fate determination seem to be preserved during evolution, one might expect that the control of neuronal number and differentiation will be basically similar in all species.